Okay, I think we are going to get started. So I'm delighted to welcome you to the College Research Seminar. I'm Marie Harvey, Associate Dean for Research here in the College of Health. And we have people in the room who are coming in to get out of the rain. And we also have people come in via Zoom. So welcome to all of you. Those of you in the room, there's cookies and coffee and tea, so help yourself. And I'm also very delighted to introduce Ursula Ivanets. I said it correctly. That's Polish Ivanets. If you want to learn how to say it, she taught me how. And she will be introducing our speaker for today. So welcome to everybody. And I'm not introducing. So I am so sorry to her that she's going to introduce our speaker. I think I'm going to let you do that. So welcome everybody, and I'll pass it on here to Ursula.
Well, thank you all for coming to our Friday seminar. Today, we're going to have Dr. Dave Dallas do a presentation on best methods for preserving bioactive proteins in milk for babies. And a little background, Dr. Dallas is an associate professor, recently promoted in nutrition, and joined OSU in 2016. He received his PhD in nutritional biology with an emphasis on biotechnology and analytical chemistry from the University of California, Davis. He also did his postdoctoral fellowship at Davis, but in food science and technology. So he's got multi-dimensional aspects in both nutrition as well as food science. His research focuses on improving health outcomes in premature babies, and premature babies are at risk for higher mortality, higher morbidity, because they're not fully developed yet. And Dr. Dallas focuses on two aspects of improving the health of premature babies. He focuses on milk, as well as what happens to the milk, the compound in milk, as they are digested by the babies. So, and again, milk, it's the wonder food, carbohydrates, proteins, fats, etc. But it also has a lot of bioactive components. These are peptides, some of which, for example, have antimicrobial properties, and he's interested in identifying these. And then he's also interested in how these peptides function in the gut in both premature babies as well as full-term babies. So his research transcends babies, across both premature and mature. In terms of productivity and support, he's been very productive from the start at OSU. Currently, he has 84 peer-reviewed publications, over 3,000 citations with an H factor of 31. That's exceptional for where he's at in his field. And his research has been funded by close to $17 million over the last 7 years, and now a little bit into the future. And he gets funding from the NIH, the USDA, various foundations like the Melinda and Bill Foundation, as well as from private industry. So endowment for research funding. And before I let it talk, I just want to indicate that for those of you on Zoom, you're unable to communicate with us except for the question and answer session. So the question and answer feature. So please write your questions, and then Suzanne and I will read them to state it either during, you want to have questions during the seminar or wait until the end. During or until the end. So I'm going to present David. Thank you.
Thanks, Ursula. That was a really nice introduction. Appreciate it. Okay. So, that thing. No. Okay. There we go. All right. Well, so Ursula introduced my topic really nicely. And in general, I have a lot of research on human milk, also cow's milk, what happens inside the gut for proteins and peptides and other bioactive components, and just in general trying to figure out how to improve nutrition and use milk as a model for improving nutrition. And so for today's talk, I'm going to take this small piece of my research, which is more recent, and it's all about how do we best preserve the functions that milk has, so that babies can get the best possible nourishment from the milk that we have. So why do I study milk? I'm interested in milk because it's the result of this evolutionary process over 250 million years, starting with very early lactation where early pre-mammals were just keeping these papery eggs moist, kind of like a platypus from these early pre-mammary glands, and then evolved, one piece at a time, adding all the thousands, millions of different compounds in milk that allowed for it to be a complete nourishment system. So, the infant can persist with just milk as their source of everything that they need, right? And the reason that that's interesting to me is because it's kind of like a tug of war between the lactating parent and the infant, because every single resource that would go into milk to improve infant health, also that comes at a cost to the mother. They have to break down their own resources, their fat stores, vitamin stores, to deliver that to the infant. So there's this competition for resources, and therefore we know that everything that ends up passing through evolution to be secreted to milk is beneficial for the infant. So that's why I'm really interested in studying milk in general. So human milk is the best nutrient source for preterm babies in particular. So what are preterm babies? So these are babies born before 37 weeks of gestational age. About 10% of all babies in the US are born prematurely. And with early birth comes immature development. So they have reduced digestive capacity. They have lower levels of different enzymes in their gut, less complete development of the intestine and the stomach. They have an immature immune system. And because of those factors, they're at an increased risk for poor health outcomes. So like poor growth is one of the major ones for preterm infants. So physicians try to feed as much as they can, but still we're ending up with poor growth in a lot of preterm infants. And then also they're at a risk for poor neurological development, and also higher risk for infections. And so compared to feeding formula, if you feed preterm babies in the hospital with human milk, especially the human liquid milk, it reduces the risk for various diseases like necrotizing enterocolitis, which is this massive bacterial overgrowth that occurs in preterm babies, cause them to die, has very high mortality rates, sepsis, other features of metabolic syndrome, and then also potentially some studies indicate that human milk leads to better neurological developments compared with formula. So human milk has hundreds, potentially thousands of different proteins in it, and these proteins contribute to its ability to provide benefits to the infants. And these proteins have an array of different functions that they can provide. So they're antimicrobial proteins that can either bind to bacteria and prevent them from adhering to the gut. So it's like a passive anti-adhesive function. There's also proteins that actively kill bacteria that can break their cell membranes, causing them to lyse. So we'll talk about these proteins a lot today, but like immunoglobulins, about immunoglobulins in human milk and how they help bind things like bacteria or viruses and help prevent them from infecting the infant. Also, another big one is lactoferrin, which is a really strong, potent antimicrobial protein in milk. There are also proteins that have enzymatic functions in milk, and some of them are digestive enzymatic functions. So there are enzymes that break down fat called lipases, and there are enzymes that break down proteins called proteases that are secreted in milk itself that actually help the infant digest the milk that is being fed. So these are really important. And there are proteins that assist in absorption of different nutrients. So, for example, the casein proteins, they're a major protein in milk. It's what cheese is made out of, right? Casein. They help with absorption of calcium. They're the protein lactoferrin that I talked about that was antimicrobial. It also helps with the absorption of iron in babies. And there's other proteins like alpha-lactalbumin and transcobalamin that can also help increase absorption of minerals and vitamins. And then milk also has an array of different hormones that can help with guiding the development of the infant. So it has things like transforming growth factor beta and insulin-like growth factor. Actually, there's a list of like 100 different hormones that are in milk that could influence development in the infant. And then there are a very large number of immune active proteins in milk. Besides the immunoglobulins I talked about, there's a huge array of different cytokines, which are like cell mediators of immune function, and various other aspects that are also important for the immune system. So preterm babies are very often, especially in the neonatal intensive care unit in the hospital, are often fed donor human milk. And the reason for that is that most often preterm the parents of preterm infants can't produce enough milk to fully feed their infant. There's a lot of different reasons for that. Early birth can kind of mess around with the hormones that are needed for a large or a high level of lactation. There's stress involved that can decrease lactation, many different factors. But the bottom line is that over 60% of parents who intend to feed just breast milk can't do that because they're unable to. So human milk banks have arisen to fill that gap because we've learned how important human milk bank is for the health outcomes of babies. We know that feeding formula is not a great idea, especially for the necrotizing enterocolitis that I talked about, this gut disease. And so there's a ton of efforts to make sure that babies are getting all human milk diets, especially up until like 36 weeks of adjusted gestational age. And yes, and most babies in the preterm infant care unit are fed at least some donor milk. So the problem comes along in that, so donor milk is milk donated from other lactating parents, other mothers who are producing excess milk, right? And they're donating these banks. The problem is that preterm infants are very susceptible to disease, infections, that sort of thing. So we have to ensure that that milk is safe for them. And so there has to be methods in place to eliminate bacteria and viruses that could be causing infections in infants. So the most commonly used methods are heat-based. Actually, all the ones that are in current use are heat-based, and the most common of those is Holder pasteurization, which you can see here. It's basically just a water bath where you're just heating milk to 63 degrees for 30 minutes, just low temperature, long time pasteurization. And that's used by basically all nonprofit milk banks. And then there are some more intensive methods. One is called ultra high temperature short time sterilization. And this technique actually sterilizes milk so that it can be shelf stable, i.e. you don't have to store it in the freezer. It can be stored at room temperature. There's a company, a human milk company that uses this technique. And then there's retort sterilization, which is a high temperature, 121 degrees for a pretty long time, 5 minutes, with added pressure. And that is also another technique for sterilization to make something shelf stable. And another large human milk company uses this process. And all of these different processes are currently used for the products that are being used today in neonatal intensive care units in hospitals. So the question that we had was, do these processes affect the structure of the bioactive proteins in human donor milk? What is the result of these processes? So our study, we designed a study to basically test all of these different factors. So we're comparing raw milk to frozen and thawed milk to low temperature, long time pasteurized, and ultra high temperature and retort process. And then we also added with and without homogenization. Homogenization, you probably drink homogenized milk, right? It's a process where you basically spray milk through a very fine needle with a little bit of pressure, and it causes milk fat globules to break and then coalesce at smaller sizes. And it makes it so that milk fat doesn't separate to the top of your milk. So like when you have milk at home, like the cream doesn't rise to the top unless you have unhomogenized milk. So some of these commercial milk banks also use that process because they want to have their product look uniform. So that's another factor that could damage proteins. So we did a simple gel just to look at the overall protein profile of these different treatments. So from here, when you see bands disappearing, it means that they are getting denatured and precipitating out of solution, which means they're ostensibly lost. So we can see that like the ultra high temperature sterilization and the retort processing cause a lot more losses of these protein bands than the what's called vat pasteurization, which is another word for Holder pasteurization, but it's a scaled up version. So it's done on a much larger scale. But same idea, 63 degrees, 30 minutes. Oh, and Celsius. The temperatures are in Celsius, not Fahrenheit. So then we used another technique to look at some specific milk proteins that we were interested in. It's called enzyme-linked immunosorbent assay, or ELISA. It's a common technique in biology. And basically this has two antibodies that are specific to a protein to detect whether it's still present in the same form, in the native form, basically. So this technique was being used. And so I'm going to tell you about the effects on a bunch of different key bioactive proteins that we observed. So for example, with immunoglobulin A, which is maybe one of the most important proteins in human milk for immune function, we can see that freezing and thawing had a little bit of reduction compared with the raw. And we also saw it's pretty much stable with the freezing and thawing for vat pasteurization, which is like Holder pasteurization. So some reduction, but not too bad. Whereas we saw a complete removal of this protein from solution through retort processing and ultra high temperature processing. So those techniques that are being used are basically not delivering any active immunoglobulins based on this method. So similarly, another immunoglobulin called IgG, we saw actually pretty good preservation of it with freeze thaws and vat pasteurization, but total removal of it with the higher temperature retort and ultra high temperature processing. Similarly, IgM, which is a more complex antibody that's common in milk, we can see preservation of that with Holder pasteurization and freezing, but loss of it with retort and ultra high temperature. One of the key proteins that are helpful with protein digestion in babies, which is called elastase. This one, we actually saw that any homogenization or any heat treatment completely wiped out its levels. The only thing that preserved it was just regular freeze-thaw cycles. So any amount of heat or homogenization added destroyed this enzyme. Then we looked at lactoferrin, which is that key antimicrobial and iron-binding protein. And we saw there is reduction with older pasteurization that we can see. And there's more reduction with the retort and ultra-high temperature processing. Lysozyme is another antimicrobial. I just put this here as an example because some proteins aren't affected by heat processing. So some are really resistant. So this antimicrobial protein, it doesn't matter. We could treat it with very high levels of heat and it still survives. So proteins totally vary in their sensitivity to these heat treatments. We also checked out the vascular endothelial growth factor in milk. It's a factor that might be modulating intestinal development in infants. And we saw good preservation with just homogenizing and thawing and vat or holder pasteurization. And then we see large decreases with retort and then somewhat lesser decreases with ultra high temperature. And the story is basically the same here for transforming growth factor beta, which is another immune development regulating peptide hormone. So overall, with the ELISA results, we saw that the vat pasteurization technique amongst the different heat processing led to better preservation of some key bioactive proteins like immunoglobulins, lactoferrin, polymeric immunoglobulin receptor, vascular endothelial growth factor, transforming growth factor beta, compared to the retort and the UHT processing. And preservation of these proteins might contribute to the improved functionality of milk in infants because they have functions like antimicrobial, effects on protein digestion, immune modulation, and intestinal development. However, these functional attributes have not actually been compared in infants yet in a human clinical study. So we know that holder pasteurization has less benefits than raw milk, but no one has actually studied these products that are being currently used to feed babies, whether they actually affect infant health outcomes between these different types. So we've also looked at this using a proteomics method. So this is a method that allows the measurement of a large array of proteins using what's called mass spectrometry, which is just measuring compounds with very fine sensitivity in terms of their mass so that we can determine what they are, basically. And so this approach, we're isolating these proteins that are soluble, that are non-denatured, and then we are preparing them for mass spectrometry and analyzing them. So overall, if we look at the overall abundance of all those proteins that we identified, we found that VAT PT is no different from raw in terms of the overall total intensity of these proteins, but that UHT and retort processing starts to decrease those numbers. And then if we look at individual proteins, so this is a heat map and it looks crazy, but it's actually a very powerful image because so in the center, we have the raw milk. And you can see most of these lines are bright green. Okay. So these are based on Z-scores. So this means things that are green are approximately 3 standard deviations higher than the lowest abundance of this particular protein across the sample types. So these are the hundreds of different proteins lined up here, and each one of them across the sample types is shown. So what we can see here is that the raw and the vat pasteurization or holder pasteurization are most similar in terms of the overall protein abundances compared to each other. And as we move away from this, we get much more black and red, which is lower abundances with retort processing and with UHT processing. And then even lower levels when we move to retort processing with homogenization and UHT processing with homogenization. So these steps are not just degrading the few proteins that we measure by ELISA, but are degrading like hundreds of different proteins that we can measure by proteomics. And this is just another way to visualize some of these changes. So these are called volcano plots. And so, for example, we have raw versus vat pasteurization or older pasteurization. And we can see there are some proteins that are significantly reduced in vat pasteurization compared to raw, which is what you can see sort of in this left quadrant. But if you look at raw versus ultra high temperature here and raw versus retort processing here, you can see there's many, many more proteins that are significantly reduced and by large fold changes than there are for older pasteurization. So it's just another indicator that there's a lot more changes happening with these higher temperature processing methods. And this one, I apologize that I didn't add these statistics here, but if you compare the proteomics to the ELISA, you can see very similar results. So the same kinds of things that we're observing decreasing with ELISA, we're also observing it with proteomics, which helps to verify the findings. So summarizing those results, we have found that pasteurization allows for better preservation of human milk proteins than ultra high temperature processing and retort processing by both ELISA and proteomics. And I think it's really important for the nonprofits and companies who are handling human milk for donor human milk, for them to think more carefully about how the processing that they're using affects the bioactives in milk. They may be choosing options that are just easy or provide a long-lasting product, something that can be stored on the shelf so it's less expensive to store, but they're not considering the impact on the actual babies who are going to be fed.
You indicated that the vat pasteurization is the most common one, right? So
yes. Yeah.
From that perspective.
Yeah. So all the nonprofits, milk banks are using this vat pasteurization method. Yes. And then the other thing is like clinicians need to think about how these different products are created before they choose what to feed, because clinicians make choices about which products to use. So great, we found that older pasteurization is better than those other techniques, but even older pasteurization degrades some proteins. One of them, for example, is called biosalt stimulated lipase. It's one of those enzymes that helps babies digest. In this case, it helps them digest fat. And this enzyme is produced, secreted in milk, and it's turned off. And then it only turns on when it enters the baby's intestine, when the baby secretes biosalts, and that turns this enzyme on. So it's this interesting enzyme that has to work with the milk and with the baby, right? And unfortunately, all the heat treatments, even the low-level vat pasteurization, completely destroys this enzyme. So that means the baby is not getting as much help in fat digestion as they would with raw human milk. And this is really important because fat absorption is essential to babies, because fat is the highest caloric component of human milk. And obviously, they're having poor growth already, so we need to make sure that they're getting all the calories that they're being fed. And there was a previous study where they fed pasteurized parents' own milk versus raw parents' own milk. And they found that with raw, they had 20% higher fat absorption than pasteurized milk. And there's other studies that I didn't list here that indicate that donor milk versus raw milk leads to poor growth outcomes and length growth as well compared to raw milk. Yeah. So we're interested in how we can make donor milk safe, but without compromising its benefits. So basically, can we think of a method that could be better than older pasteurization? So we were looking into two methods in our lab. So high pressure processing and Ultraviolet-C light. So for high pressure processing, so this is a common technique used in the food industry for different products. So in general, you pressurize the food from 100 to 800 megapascals. Basically, you're squishing water in a container to make more pressure added to the products that are inside that water. So you're just like squishing these bags, right? And that squishing distributes that higher pressure within the liquid, and it causes the cell membranes of bacteria to break. It's like a change in pressure, right? And this causes changes in the structure of the DNA, which can prevent bacterial replication, and it can also inactivate some viruses. So we designed a study to look at what would be the optimal processing methods for high pressure processing. So we didn't know exactly what settings to use, how much pressure, how much time should be applied to kill enough bacteria. And so we tested all combinations of these pressures and times on aliquots of a large pool of donor human milk. And then we compared that with no treatment. And we tested how did that affect the survival of pathogenic bacteria that we spiked into the milk prior to processing. So we tested both vegetative bacteria and spore-forming bacteria. And then we tested how that affected the proteins via ELISA and proteomics. So from the the vegetative bacteria perspective, we were able to find some settings that allowed for at least a 5 log reduction. 5 log reduction, it means 99.999% reduction of bacteria. And that is what's required for like cow's milk processing. And that's the standard that's used for human milk. An unofficial standard, it's not regulated governmentally. But we found that 500 megapascals for 9 minutes was enough to provide this reduction of bacteria for the bacteria that we tested, including some that are quite pressure resistant. And these are bacteria that are commonly occurring pathogens in human milk. However, we found that when it comes to spore-forming bacteria, these are like the senescent forms of bacteria. They're much more resistant to degradation. Pressure is not working very well. So we got about a 1 log reduction of these bacteria. So this is a bit of a problem. But if we compare to Holder pasteurization, that also doesn't eliminate spores, and it has to be stored frozen. It's not a shelf-stable product. So this is basically as good as Holder pasteurization from a bacterial perspective. But we would like to be able to eliminate spores as well. So then we repeated what we did with the previous treatments, and we found that when we look at these particular proteins, like the biosalt stimulated lipase, we did an activity assay, and we found that here, Holder pasteurization completely wipes out this enzyme, but we have good preservation of this enzyme across all different levels of high pressure processing treatment. So good, that was a good result for us. We found Holder pasteurization has this somewhat of a reduction on immunoglobulin A, like I talked about, but we see better retention of it with high pressure processing. Similarly, we found improved retention of these proteins with high pressure processing for lactoferrin, immunoglobulin G, elastase, immunoglobulin M, and polymeric immunoglobulin receptor. And we have also been investigating Ultraviolet-C light. So UVC light is another non-thermal approach, so no heat is specifically added, and it can be used for bacterial and viral elimination. So it's often used in water treatments. And basically what's happening here is that the UVC energy that's released from the lamp is absorbed by DNA in the viruses and bacteria that can cause basically structural changes that can prevent them from replicating. Oh, and UVC light is at a wavelength below that of visible light here. So this is just an image of, a potential mechanism that could be occurring. So UV light is causing this cross reaction between bases in the DNA, which prevents it from them being translated correctly for replication. So we did the same kind of method that we did for high pressure processing, but with UVC. And in this case, we use a very simplistic model of this. It's basically just a UVC lamp stuck into a beaker of milk and with a stirrer. So it's just like stirring really fast. The stirrer is because milk is very opaque. It's white, right? So it's hard to get UVC light to penetrate through it very well. So we need to stir it to make sure that everything is getting close to the lamp, basically. And we found that in this section, we've got our vegetative bacteria mostly. And we found, a dose of 6,000 joules per liter of light was enough to get a 5-log reduction of all of our vegetative bacteria, which was great. And we found that if we increase the dose, we were able to get a reduction of all the spore-forming bacteria as well that we needed by 12,000 joules per liter. So about double. So that's great. That means this could be used to make shelf-stable product as well as a refrigerator frozen product. So we looked at some different proteins by ELISA, and I just simplified this graph to look at only the treatments that were the optimal ones. But we can see here that we have, some losses of IgA with HOP, but full preservation of IgA with UVC, which is actually a better result than we had for high pressure. Same with IgG, full recovery of IgG. No statistical difference for IgM. Same with elastase, cathepsin D, and lactoferrin. So we found better preservation of all these key bioactive proteins with UVC than high pressure processing, and both were better than Holder pasteurization. We also did some activity assays. So these are like enzyme activity assays. And again, we found great preservation of the bile salt stimulated lipase compared to the complete loss with Holder pasteurization. And this one, we found preservation of lysozyme activity as well. From a proteomics perspective, remember these volcano plots. So this one's kind of more zoomed in than last time. So we can see some more proteins that are reduced from vat pasteurization or Holder pasteurization. And we see fewer of them that are reduced in UVC at, this is 5,000 joule per liter, and fewer also at 11,000 joule per liter. So it suggests that the overall protein profile is less reduced by UVC than Holder pasteurization. So then we are working on designing a novel device. This is with Adam Higgins in bioengineering. So this is a microfluidic chamber. So it has a very narrow chamber, just 100 micrometers in depth. And this depth is designed based on how well UVC can transmit into the opaque milk. And so this depth is enough for it to transmit all the way through and reflect back off of the plate. So we have good good introduction of the UVC light into the full milk as it flows through. And it's a flow system. It's being pumped through this chip, and it has LED lights that are UVC wavelengths shining down at the milk as it passes through. And so far, what we found is that this system allows us to get the 5-log reduction of these pathogens that we needed, both spores and vegetative. And interestingly, with this system, we're able to use almost a 4x lower dose for vegetative bacteria and almost a 7.5 x lower dose for spore-forming bacteria. And this is really important because by lowering the dose, we lower the risk that the UVC light could alter other things in milk. One of the issues with UVC light is that it could damage vitamins in milk, like light-soluble vitamins, light-sensitive vitamins, and it might damage lipids as well. So the lower we can go with the UVC dose, better. And I'm working on an NIH proposal right now to look more into this device, and we'll be planning to look at the effects on the light-sensitive vitamins and lipids as well, in partnering with the LPI and Mass Spec Center. Okay. So then, kind of the final question, one of the final questions that we have a lot of questions, is will improving preservation of these milk proteins make a difference for infant health outcomes, right? That's important. So we are doing a feeding study right now where we're feeding colder pasteurized milk and high pressure processed milk to preterm babies in the NICU. Sorry, Ursula, what does that mean?
Well, I'm worried.
What's my deadline?
Another 10 minutes.
Oh yeah, I'm good. Okay, sorry. So we're doing this clinical study right now. So we've got enrolled the same babies, and it's a crossover design. So they're fed colder pasteurized milk one day, and then we collect stools, and then they're fed high pressure processed another day, and we collect stools. And then we've collected from 40 babies so far, and we're basically taking the stool samples, and we're going to measure the total lipid contents that are in stools. And what we're trying to do is try to figure out the percent of fat absorption that these babies had. And so the real specific question that we're trying to ask is, because high pressure processing preserves this lipase that I talked about, the biosalt stimulated lipase, does that preservation result in higher fat absorption for these babies, right? That's the specific question. So we'll be using LC-MS to quantify all the fatty acids that are present in those samples to do comparisons.
Did you say 4 or 40?
What?
Did you say 4 or 4?
Oh, 40 infants. So we're at the stage of doing data analysis.
Okay.
So,
okay.
Yeah. Okay. So just summarizing the main points I made today. So as you're well aware now, milk provides a lot of different functions, and these are beneficial to the infant. And how human donor milk is processed really highly affects how milk proteins are preserved, and probably how other milk components are preserved as well. And we found that colder pasteurization was superior to ultra high temperature pasteurization and retort pasteurization in preserving key bioactive milk proteins. And we also found that high pressure processing and ultraviolet-C light irradiation was even better at preserving proteins than colder pasteurization. And we do need more research to optimize these methods further, and also to define how they affect the whole host of other components in human milk, because it's not just proteins that matter, right? I care about proteins, but we need to be more broad, because clinicians, milk banks, they need to know this information. And processing can likely affect infant health outcomes, right? That's the final thing. And we need to do that research to figure that out. And I think it's important that this research can be used currently to help guide milk processors in how they produce milk, and clinicians in how they select these products for infants. And just want to acknowledge my group. I didn't do any of this work myself. It's all my grad students and postdocs and collaborators. A lot of collaboration that you saw here today was through food science from Joy Wait Cusick's lab, looking at bacterial reductions, and Adam Higgins, who designed the UVC system. And mass spec work is done at the OSU Mass Spectrometry Center. And then all the infant sample collection is done at Oregon Health and Sciences University with Brian Stotolini. And some of the processing was done at UC Davis in their milk processing laboratory with Daniela Brile, and some at North Carolina State University with KP Sandeep. And then all the milk that we studied, and this was hundreds of liters of milk, came as a donation from the Northwest Mothers Milk Bank. So we have a really good working relationship with them. Okay, so if you have any questions, I'd be happy to take them. So thanks.
Well, thank you so much. Questions? Marie?
I don't really have a question. I will, but I think about it more. But what I really want to say, I really appreciate how you presented today, because this would have not been understandable if you hadn't so carefully outlined it, defined things as you went through. So it must be a great teacher.
Oh, thanks, Siri.
Thank you.
Thank you. Yeah, I was trying to make sure, because I know we have a very diverse audience here.
So it was well done. Thank you.
Yeah.
Laura?
I echo that because I totally understood it, and I didn't think I would. But I really appreciate also your attention to public health and like just really looking at the health benefits, especially this very vulnerable preterm babies is so impressive, and also all the collaborations. And I was just curious because you said it was so important to inform this variety of systems. You said there's NGOs and others that do this preservation. So does this not fall under any regulation like FDA or anything? Can you speak to, or if it doesn't, as your work seems very important perhaps for informing policies that do provide the protections that also is overall comprehensive health promotion because of preserving those proteins you talked about.
Yeah. Yeah, no, that's a really good question. So basically, human milk banking is not regulated the same way that bovine milk or, other animal milks is. So like, those animal milks that adults consume, there's very defined regulations, like has to be exactly this temperature for exactly this time. And there's, all this testing that goes into that. Human milk banks just use a voluntary system. So they're not regulated by the government. They just decided to use a system that matches what they use for bovine milk. And, there's just not I guess the point is they're just trying to be as safe as possible, right? So they're using these systems that are in place. And then, yeah, like what you're saying, like in terms of the public health guidance, there is no guidance out there about like optimal processing for human milk. There's only research papers, really. There isn't any sort of government documents about it. So it could be something to go towards in the future.
Yeah, it does seem like you have, since you were so great and articulate in describing your work to us, that translation of your science for these populations, the NGOs, even mothers, understanding, I don't know, it seems very important.
Yeah.
So that's great work.
Yeah. Thank you. Yeah. I mean, I try to stay really connected with the milk bank here, the local one, just to try to like think about what kind of improvements they might take on, basically. So yeah,
That's awesome.
yeah. Thanks.
Thank you. Really, really interesting. And I love where you go from, really detailed lab work out into human research. So that's really wonderful, because that was one of my questions when I was listening. It's like, what's the next step? But I have a question that kind of follows up on what Laurel said, which is around consumption, the production and consumption of human milk for these infants. How receptive do you think the for-profit organizations are that are using these, high temperature processes will be to information of this sort that suggests? And maybe this is totally outside your realm, but
No, it's okay.
to me, yeah.
Yeah. So, when a company has a production scale facility and has invested a lot of money to developing it, I'm sure they are quite reticent about like changing their process, because it would be very expensive. So I don't know. I mean, mostly what I see in the industry space is that people are just doing what they decided to do and just promoting their milk based on like, what it is, and not as much like innovation in terms of new ways to improve the product in terms of like the bioactivity. So yeah, so not as much there really.
So the angle might be to go on the demand side, which is if the hospitals and clinicians are saying we only want this particular process, you could maybe shift perspectives out.
Yeah, I think so.
Mothers and mothers, sorry.
There's also like a cost part of it too. So like the companies that are selling the sterilized versions, they're selling it for a lower amount of money because it can be shelf stable. So that's part of their scheme, is like get milk to more babies. So yeah, there's a lot of factors here. Yeah.
So how many milk banks are there in this country?
Oh,
We trying to have some.
no. Oregon has one milk bank. And I think, and that milk bank is for Oregon, Washington, Idaho. So I think there's probably about like 10 or 15 milk banks in the US. So they basically serve as like a central repository. So you may drop off your milk at various different pickup sites, and then they're shipped on dry ice to these sites, and then they're all done. The processing is there. And because they have to do a lot of testing at those sites, they have to like monitor the mother's blood for viruses. They have to check health status, all these different things, right? So not that many of them. Yeah.
Needle? Yeah, CJ. Can you comment, or maybe somebody else in the room knows, what is the, human milk changes from distance from birth, right? So an individual mom would not likely have just given birth to a premature or newborn. Do you have an understanding of what age distance from the original donor on average we're talking about? And do you see that at any point being some kind of confounder that sampling and testing would need to consider the age and the different fat component of that milk when comparing to?
Yeah, that's a really good point, Neil. So yes, you're totally right. The lactating parents who are donating are much further on into lactation, usually. So usually like 6 months into lactation, where they've got this whole thing going really well and they're producing extra. That's typically when people are donating. And so, yes, you're right. Milk changes across the time of lactation. So the milk that they're getting is different than the milk that they would be getting from their own lactating parent.
But if you were doing your analyses across the same similar, I'm saying age of milk, but I mean since that donor's birth experience, there should not be a confounder there. Was there any attention to that, or is it not likely to be a big player?
Well, so for our study, we thought about that, and we used one large pool of milk from a lot of different mothers.
Yeah.
And so we controlled for that. But other kinds of studies in literature, there is that effect that can be occurring for sure.
That seems reasonable.
Yeah.
Thanks.
CJ, and then if we can go all get it.
I do agree. I don't have a good history background, so I was like really impressed with how you described it. I found that it's done worldwide, like even Iran is I'm like pretty amazing.
Yep.
So what my question is in terms of time, like you said, there's a higher cost with the UVC. Is there does it take longer when you're doing it compared to the other?
Oh, yeah. So I guess two parts of that is like, one is cost, and two is how like the productivity basically, or the throughput. So in terms of cost, a high pressure processing system is probably not something that a small milk bank could have on site. Typically, like the kind of processing equipment that they use might be like less than $100,000 or something. I don't know, maybe less than $500,000, right? But a single high pressure processing unit might cost like over $500,000 or $1 million, right? But there are full processing facilities for high pressure processing throughout the country. So like for our feeding study, we took it to this unit that is used for a large variety of foods, like cold pressed juices that you might have seen on the market, or various different products. And so you could take the samples there to be processed in bulk, and then delivered to the hospital. So it's a feasible process. For the UVC, UVC is actually very inexpensive. Also has a very low energy cost. So this is quite feasible. Even the method that we're using with the little chip can be scaled quite easily and inexpensively to meet the exact same volume processing demands of a milk bank. So, yeah.
It's a comment from an initial CR, hmbana.org, links to the Human Milk Banking Association of America. Sharing.
Oh yeah. So the Human Milk Banking Association of North America is the sort of parent organization that all of the donor milk banks in the U.S. Kind of work underneath, basically. So they provide the sort of guidelines and regulations for how milk is handled. Yeah.
So the supply meets the demand
For donor milk? That's a good question. I think in general, like, okay, so I'll speak from experiences. I don't know like the statistics, but like when I was on the board of the milk bank here, and there was often the case that they had more milk than they needed to go out. So, but I think part of that is because there's a high cost for delivering donor milk to babies. And, like, so some of it is paid for by insurance in the hospitals, but only up to a certain age of gestation. So there's restrictions on it. So it seems like it's meeting demand.
I think it's
Because of the cost.
last time I checked was like $4.
Yes, per ounce. Yeah.
So it's quite expensive.
Yeah.
Any other questions?
Dave, this is remarkable. Thank you so much. The thing that I was so impressed by was the just how the immunoglobulins were affected by the three processes. So the ultra high temperature and the retort process. The antibodies are gone, basically. No IgA, no IgG, no IgM. And did you expect that? I mean, that is that as clinically relevant as I would think? Because I mean, when we talk to people about the benefits of breast milk, that's one of the big things is, well, you're going to be able to protect yourself from diseases because all of these antibodies are there. So just curious.
I honestly didn't expect to see as much reductions from those processes as we found. Yeah, I mean, to have like non-detectable levels is really surprising. And yeah, it makes me think about, all those products that are out there and like what they're actually providing. I guess they are still providing like the same calorie source, the same macronutrient distribution, but not the bioactive component, So, and maybe I don't know, they might even be digested differently or getting into the baby differently. But yes, definitely no immune IgA function for sure. Yeah. And yeah.
This is crazy. Just Ryan's question made me think because they are premature. They're in a very controlled environment. And maybe the benefit is getting all the things that you said they may be still getting at that stage. They just need to eat and such. That may be all the immuno stuff because they're in mature. So anyway, it's interesting to think about like the
But they're also at risk for sepsis.
Yeah.
And so
Yeah.
it'd be nice to have IgAs.
No, I'm not saying. I was just thinking. Anyway,
No, yeah. So yeah, they're in incubators, but it's not like a germ-free situation. So there's a lot of bacteria, like dangerous bacteria that circulate in like neonatal intensive care units.
that makes sense.
So that they have to be
Is-
careful about.
is very important.
Yeah. Yeah. So thank you. Thank you all for coming.